Although I have a computer science background, but I was always fascinated by biology. So at some point, I decided why not to switch to bioinformatics because it's very exciting field, and I'm grateful for Alexander that he had lecture before me because he nicely showed many different applications of bioinformatics for different types of research. And today I have quite a, I would say, general lecture on the metagenomics application. It's not focused on one specific study.
Alina, sorry, I'll
But
interrupt you for a
yeah.
second. You didn't give me a chance to like even present you. So if
Ah,
you just
sorry.
no, it's fine. Just you started presenting yourself already. I'll just add a few words that Alina works at Kiev Academic University at the Laboratory of Data Science and Machine Learning. She's also the Junior Research Fellow at Institute of Molecular Biology and Genetics of National Academy of Sciences of Ukraine. And yes, she participates and leads a lot of cool biological different branches of systematic biology and other branches of biology. So yeah, please, word to you.
Thank you so much. Sometimes too different because I feel overwhelmed at times. Luckily, I have a very nice support from the colleagues and students because I was lucky enough to have very talented people to work with me. So I will talk today about metagenomics application. This is rather a general lecture, and I have some beginner's slide for gentle introduction into the topic because I expect that we have very different people today on the call with different backgrounds, and I'm not sure how people are familiar with molecular biology. So I wanted to give a super basic things first to give you this feeling of the complexity of biological system, because in many cases we operate with genetic code, which we can decipher with sequencing that was already mentioned for many times during the previous lectures on the school. But like, as we are the organisms, as a human, we consist from organs, which consist from tissues, which consist from cells. And inside cells, we have a chromosome, which is actually a compactly stored DNA, very famous molecule that stores our genetic information. It needs to be stored compactly because if you make it like a linear structure, it's quite long actually. And the genetic code it's quite simple. We have like 4 nitrogenous bases, and they actually complementary between each other. That's how DNA forms this double helix structure. And in many cases, the work of a bioinformatician to do something with these letters when we have long sequences of bases, and we need to understand what kind of organism it is or what it does. And actually, there is a flow of this genetic information in organism, which is called central dogma of molecular biology, because to be honest, the bioinformatician, what we work with the data, it usually is produced by the methods from the molecular biology. And we need to understand how this information works in a cell and in an organism, because if you want to be a good computational biologist or bioinformatician, you must understand these concepts and you must work with biology all the time. So we have this, like, this is the classical dogma. We have almost unilateral flow of the information from DNA to RNA to protein, which is called transcription and translation. However, with the development of different experimental techniques, we understood that this scheme is too simplified, and one can even create something like this, which is more convoluted, and it actually led to creation of the whole branches of science like epigenomics or epitranscriptomics or epiproteomics, because we have not only just a DNA, we have some modification to these molecules that may silence some gene expressions or the activity of the genes. We have then modification of the RNA structure of proteins as well. And this complexity of the internal machinery in the cell actually enables such a biological diversity that we can see around us. And in terms of sizes, this is also very interesting. We know that like humans, we can see with naked eye, but there are many, many more organisms that we need special equipment to basically identify. Something super small can be identified with electron microscope, and something with the light one. Some cells we can actually see with naked eye as well, but in many cases it's just something that is hidden from us, and we need special techniques to be able to decipher this information. This is actually quite interesting plot. I was searching for something like this for this lecture specifically because I wanted to give you a feeling what surrounds us. And this is how basically our understanding changed throughout the years, because for many decades we thought that animals are majority of the species on Earth, but it's no longer true. Nowadays we think this is actually bacteria, so the microorganism. And this is funny statement from this paper that actually why we thought that like animals are majority, because insects are also part of this domain. So majority of these animals, this blue part on the pie chart was actually insects. But if you try to study insects with the modern technologies like sequencing, you can actually sequence gut microbiome of an insect, and you might find every time in a new insect several new bacteria. So if we assume that we have lots of insects, different species of insect, then we have much more bacteria. So that's why it's so exciting when the metagenomic emerged. We actually opened like our eyes to a totally different world, to the world of the microorganisms. And of course,
can I, since you said that you prefer questions on the go,
sure. Yeah.
So do I understand correctly that this is like estimation? This is not the sudden discovery.
Exactly.
It's just reconsidering of how it should be like in general structured.
Yes, exactly. It is still an estimation because we are still far from identifying all the species on Earth. I'm not only talking about bacteria. I will explain later why it is hard, but even for the animals that we can see with our eyes, it's still extremely hard. I don't think we even halfway to the identification, and this is a very dynamic process because some species unfortunately die out, some new species sometimes emerge. Of course, in bacteria's world, the emergence of new species is much more rapid than in big animals, basically.
It just looks like a very drastic change, like the whole paradigm shift. So it's either like something happened, or like we completely lacked the
I think sequencing happened. I think sequencing happened because just a few slides first about like sequencing. This is I mean, the classical one is sequenced by synthesis. So we have some kind of a plate, which is called flow cell, and we have oligonucleotides attached to it, and then we have short fragments of DNA, and we can also attach known sequences to each end of the DNA, which is called the adapters, which then complementary to this oligonucleotides connected to the flow cell. And then we start the process of synthesis, which basically synthesize a complementary strand for this short fragment of DNA. And how sequencer, and we do it in many repeats until we have something like this. And how sequencer identify it is that each nucleotide is actually added one at a time, and it's labeled. So every time we add something, if it attaches, because it's a complementary, it's the signal is detected, and then we basically know the sequence of this synthesized fragment of DNA. I think it was already mentioned, maybe by Alexander, that in many cases we actually, even if we work with RNA, we actually don't sequence RNA because RNA is a less stable structure. We create complementary DNA for this RNA, and that's sequence DNA. There is an exception to this, but I will explain later. So, and this is exception. Yeah, I wanted to mention the sequencing, even though Alexandra also mentioned it, because it's actually is very influential for the metagenomics. This is so-called nanopore sequencing. It's from the company which is called Oxford Nanopore Technologies, and they created a sequencer that is of a size of even less than modern smartphone. They have several variations. One was even able to attach to smartphone. Then they have smaller FLONGO, bigger grid ion, and a very, very big PROMET ion. The difference between some of them are in the inside machinery, especially flow cell, because for this big sequencer, they have more pores, protein pores, which basically allows you to make DNA go through this pore, like the motor protein and pore and then membrane, and then we detect ionic current while this DNA goes through the pore, and we get some kind of this graph, and then we can decipher the bases. And since this technology is extremely portable when it comes to mean ion, it means you can go to faraway location, take the sequencer with you, and sequence on site. Maybe for some reasons it was not possible before to transfer the sample, or a sample is too rare, or the transporting conditions are not feasible for that. And then if you can go somewhere and you can sequence on site and you can get real-time information about the species inside your sample, it actually advanced metagenomics together with this first creation of like modern high-throughput sequencing. To be honest, this is a very nice case where deep learning is actually shines brightly, because on this plot the signal is super clean. Yeah, you can just visually say that, okay, those are like different bases, and I can even write down it myself, but this is like simplified information because it looks much worse. And it's not like it's deterministic and we read ionic current for one base at a given point of time. Sometimes the signal is like blurred. If I'm not mistaken, it detects three bases at a time. I'm not sure about the latest flow cell version, because Nanopore releases new flow cell every, I think, maybe 2 years or something like this. And previously, why people were not paying attention to these technologies, that the accuracy of bases reading, or in other words, base calling, was quite low and not applicable for like human research. But nowadays it's getting there. It's getting to the level of accuracy of other sequencing technologies. So that's why people actually use deep learning in many cases to do the base calling for the nanopore sequencing, because it's very suitable method. And what you can do, you can synthesize fragments of DNA with predefined sequence. It will be like A, T, T, and so on. And when you sequence that, you know what to expect. And then you can train your network on this type of data, and then you can build basically a classifier that will output you your sequence of the nucleotides. Nowadays, there are like number of these algorithms for nanopore sequencing base calling, and I think even though they're quite accurate, people are still actively researching for the better algorithm, especially when it comes to the RNA modifications, because this technology allows you to do the direct RNA sequencing. So you do not need this additional step of like converting RNA into DNA. You can directly put the RNA molecules through the spore. And this is why also this technology is so powerful for metagenomics, because then you can study RNA viruses. or you can even go and do metatranscriptomics, which is even more exciting than metagenomics, I would say, or they actually should go together. So I should mention this anyway, because this is such a ground project for the sequencing itself, because I think it gave us lots of information. It gave us the understanding that simply deciphering the sequence of DNA genome is not enough to understand what is happening. And I think it gave a boost for sequencing technologies development, because it actually took 13 years to get full human genome, because it was so hard with that sequencing technologies that were developed at the time. And I can show you this on this plot, because here we see the speed of sequencing. And somewhere in 2007, when Illumina introduced their technology, which is also based on sequencing by synthesis, we have like rapid growth, because we finally got to the point where we have this high throughput sequencing, and this human genome project was happening somewhere around here. That's why it took so long. And this so-called third generation sequencing is actually nanopore sequencing, also PacBio, which is sometimes called single molecule sequencing. PacBio, it's a technology which also allows you to get super long DNA sequences, because for the Illumina it's usually short, and long sequences give you the possibility to sometimes capture the whole genome of microorganisms, which is extremely efficient, especially for the downstream analysis. So you can omit some steps because you already have the full genome. Especially it's also very important for plasmids, because plasmid is a short circular DNA structure inside bacteria, which actually responsible for horizontal gene transfer. This is one of the major mechanisms how bacteria transfer the antibiotics resistance genes. So that's why this part, I think it partially answers why this happened, this dramatic shift, this dramatic change in the paradigm, because we discovered whole new world. We started sequencing everything, and we started to see much more things we couldn't see before. Yeah, and of course, the price of sequencing, it's dropped significantly, and now we can afford to sequence like hundreds of samples in a single project. So this was also mentioned by Alexander, that we actually have many different so-called assays, different types of sequencing experiments that would target different types of molecules. It can be RNA sequencing, maybe DNA sequencing, ChIP-seq, ATAC-seq, mass spectrometry, because we are also interested in proteins sometimes. And some of them are widely used for metagenomics. Of course, it's shotgun sequencing, so basically DNA sequencing. But I also mentioned metatranscriptomics, and for metatranscriptomics, you would rather use direct RNA sequencing in many cases. Yeah, that's why, I mean, as a bioinformatician, I still try to educate myself in all these different wet lab techniques when it comes to sequencing. I don't do anything with hands. I don't pipette. But if I don't understand how the sample was extracted with which kit, I cannot assess the artifacts I see in the data, because metagenomics, unfortunately, very sensitive to the extraction steps, because we have many different organisms in a single sample, and sometimes you cannot come up with the optimal parameters for your extraction step to extract the genetic material in an unbiased way. This is typically how RNA sequencing experiment looks. So you would have mRNA, then you will convert it to cDNA, and then you will fragment it, because for typical sequencing by synthesis, you need DNA of certain fragments to prepare your so-called library. So a mix of original material, adapters, and everything you prepare before you put everything into sequencer. And then you would, in the best scenario, you would have a reference genome. So it serves as a template, it's already known. And then you will try to map these short sequences to this reference genome. In this case, we also deal with so-called like exonic regions, intronic, because for the regular gene on a DNA, it usually has structure, because gene, to be honest, gene, this is how I define this. It's quite an abstract definition, because we deal with transcripts, so the product of the gene, if you can name it like this, and from the single part in the DNA, which we call gene, we can actually get many different transcripts, and we can get many different versions of this gene, and then they can also be translated in a slightly different versions of proteins, or maybe sometimes very different. And that's why we have this extreme variety of molecules in a cell. And for each of these molecules, we would have this specific assay to capture it. Okay, and how exactly it's different to the rest of like typical sequencing, or maybe I would say rather the difference is in the downstream analysis, because this is second part of answering your question, why we had this paradigm shift. Because the classical way to study bacteria would be to culture bacteria, so to grow it in a petri dish. But to grow it, you need medium, so basically nutrients that bacteria would feed on, maybe some specific temperature for this type of bacteria. And unfortunately, in many cases, those parameters are unknown, especially if we deal with completely unknown bacteria. That's why if you try to grow something, maybe some random sample you collected somewhere, you will see something, but what is there? I mean, everything survived, or maybe there was some dominant bacteria which is not very sensitive to the conditions you had within your petri dish, and it's just grew, and then you basically you cannot assess what was there in the beginning. But with sequencing, we can completely omit this step, and we just can sequence everything we have in the sample. So strictly speaking, I'm talking here in many cases about bacteria, so maybe viruses, because metagenomics firstly targets microorganisms. But to be honest, when you sequence your sample, you will get everything that was in your sample. So if for some reason, maybe you collect sample from the soil, you take soil and then you extract DNA and you sequence. If there was DNA of a plant there, you will also sequence it. If there was a DNA of some maybe small eukaryotic organism, it will also sequence it. It might not be the best tool to study plants, but at least you can get variety of organisms that are in the sample, actually including human, if you, for some reason, the human DNA happened in the sample. And but I will rather focus on bacteria than archaea and part of eukaryotic organisms such as fungi or maybe like some super small single cellular organisms. I'm not sure if you're familiar with the story of the archaea discovery, because it's actually a recent story. I mean, this tree is being updated. I mean, I think it's 1996 for this picture, but I think the recent one is already updated. So people previously thought that archaea is actually part of bacteria because structurally speaking, it's more similar to prokaryotic organisms. But then they found out like metabolic pathways in these organisms that make it more similar to eukaryotic organism, and they changed this phylogenetic tree of life on Earth. And it happened only because people were able to sequence metagenomic samples, because it was impossible to culture some things and to grow and then to study it like more precisely. That's why it's a very fascinating field of study. It's quite dynamic because you can discover something new almost every day if you sequence enough, of course. And yeah, these microorganisms are extremely different. This includes bacteria, viruses, some fungi, and as I said, like for prokaryotic organisms and for archaea, they are quite similar in terms of structure. I mean, this nucleus absence, but still, I think the modern understanding and this archaea is a little bit more close to eukaryotic organisms. And some of these organisms are, they inhabit human body. And this is also fascinating because we live together with organisms. It doesn't mean that bacteria is necessarily bad, because you all know we have gut microbiome. If something happens with your gut microbiome, you feel quite sick. That's why we need to study it, not only to defend against bacteria and viruses, but also to protect ourselves with the help of these microorganisms. So the typical metagenomic experiment is different to maybe RNA sequencing or DNA sequencing of a single cell line or tissue, is that we will expect to have some kind of a soup of or a mixture of different DNA of different organisms. But when we sequence, of course, we don't know it. We just get this pure information of the bases, and we get information in literally in this way. But it will be millions or sometimes billions of these sequences, which you need to process, and it's of course impossible to do by hand. And it adds a level of complexity because you need to have techniques how to do this reads classification. So read is this short fragment of DNA that we get after the sequencing. So you need to sort or classify this genetic information, and you need to tell this, okay, that this reads, they go to bacteria, maybe these reads, they go to virus, and so on. And then after you manage with this classification, you can do a multiple number of analysis. So the easiest one is taxonomic profiling. So basically, we just try to understand what is in the sample. Then maybe you want to focus on viruses, or maybe you can do functional profiling, which I might say is actually, in some cases, much more important than to do this taxonomic profiling. I will try to explain why. So with shotgun metagenomics, and here I specifically mean whole metagenome sequencing, when we take all DNA and we sequence everything, because there is also so-called 16S sequencing, when you sequence on the 16S region of ribosome coding the ribosome coding genes. But why this region is because it's very conservative, and to the point that you can differentiate between bacteria only using this region. So you don't need to sequence like all the genome of bacteria. You can sequence only this super conserved region and then tell the bacteria. But the it's an interesting discussion. People actually in many cases call it metagenomic as well, but it's not metagenomics, because it's not meta, because you pre-select something, because you pre-select only what is known, because you need to know this 16S region, and then you can create basically a primer which will bound to the specific to the DNA, and then detect whether this bacteria was in the sample or not. So strictly speaking, 16S sequencing is not metagenomics, but people sometimes call it still. And I think 16S is still quite a powerful tool because it's cheap. It's cheaper than shotgun sequencing. And for the applications when you have predefined list of organisms you want to check against your in your sample, this is actually the best, because again, you save some money, and then it's quicker, and then the analysis is super straightforward. For 16S sequencing, there are ready-to-use pipeline, very famous one, which is called CHIME. It's quite a nice modular system for the pipeline, and I think it has more widespread in clinics. 16S, because then you can design a very specific panel to check only for what is interesting for you, because if you studied something in advance and you know that you don't need to check everything else. But with shotgun metagenomics, it gives you much more information. As I said, it's not necessarily bacteria only or viruses. It can be anything that was in your sample. So the simplest thing we can do is marker gene analysis, because again, we have databases, we have some genes that are super specific to maybe a family of a bacteria, or even for some specific species. And then we can check whether we can find this marker gene in our reads. But even on this picture, you see that not every read would contain this marker gene. It means that in some cases we kind of wasted money, because let's say we found marker genes only in half of the reads we generated. What to do with the rest? Should we just throw away? That's why in other cases we can try to do the assembly. We can try to build a longer sequence of these reads, trying to see whether they overlap. And if they overlap, then we build contigs. This is longer sequence. And in some cases, if our sample is sequenced deeply enough, we have lots of read, we can even try to do this whole genome assembly of each or some bacteria that are in the sample, and do the binning, like to sort basically this sequences into different bins which belong to different species. And each of these analyses has its own advantages and disadvantages. Basically, this one I already said, we are very dependent on the content of the database. If our bacteria is not present in referent database, we will never find it. And for assembly, we need lots of material to do high quality assembly. And the deeper we sequence, so basically the more material we get, the more expensive it is, because like a single run of a sequencer has a specific capacity. let's say you can multiplex, I don't know, 100 samples in a single run. To multiplex, you will add different smaller adapters to each of your sample, which you can sort later on bioinformatically and assign these reads to different samples you had. But since the single run has certain capacity, you would get amount of reads per sample divided on the amount of sample you had. So if you want to sequence deeper, you will have smaller amount of samples in a single run, which will increase your cost because then you need to, for example, have 2 runs of sequencing, 50 samples and 50 samples. And this is always a trade-off. If you have a certain budget for your project, this is the first thing you consider. Whether you want to go wider and cover more cases. Let's say you have, I don't know, case and control study, or maybe you have several subcases if you're studying, let's say, inflammatory bowel disease, and which is also a hot topic because people also try to connect it with gut microbiome and so on. Maybe you want to have more patients, but sequence less deeply. So this is not a simple task to design very robust metagenomics experiment when it comes to defining what kind of extraction kit you want, what kind of library you want to prepare, whether you have like low biomass sample or not, but like whether you have lots of starting material or not. What to do with bioinformatics later is also a question, because it's actually extremely sensitive to databases. I think every year there are lots of papers released on like benchmarking different reads, like methods for taxonomic classification of metagenomics data, and they rarely agree. And people also benchmark different databases against same methods, and they also don't agree. So this is still a very challenging task. I have a very small example of how might taxonomic profile look like, and I chose this one because it's a simple one, and it's also connected to some of the previous problems I described, because we need a very strict control what is happening in our sequencing process, and we need so-called positive control. It's a sample with the known microbial composition, which you can sample and then check computationally should contain a predefined ratios of these organisms. They are commercially available, and people usually use these commercially available examples because then it's very easy to cross-compare between very different experiments. But it never works. It never works like it was designed, because here we have 3 samples, and each sample has it's a positive control, and it has 10 different species, and they need to be in certain proportions. But even from this plot you see that it's not like this. And for example, this color, it's almost not present on the first 2 samples, because this is exactly the species, this one, Cryptococcus neoformans, that it has super tough cell wall, which is it's really hard to extract, it's really hard to destroy. So you have a dilemma, like whether you go for a more harsh extraction method and basically ruin half of your other bacteria, but extract this one specifically, or vice versa. You go for gentle and you extract majority, but this one will never be extracted, and it will never happen in your resulting data set. And that's why it's really a challenge to really get a nice one. In this case, I still consider this as a very good result, because I often see worse, because this one was very, very deeply sequenced. I think maybe 200 million reads per sample, which is usually not the case for metagenomics. People don't go for such a deep sequencing because in most cases you are rather interested in the taxonomic profiles. But it's not everything that metagenomic studies, like we're also interested in functional diversity. We want not only count what known bacteria we know, we also want to identify new bacteria or maybe new compounds, new proteins, because I think drug design is also, it also gets lots of information from the proteomics data set, metagenomics data sets. And we're also interested in metabolic pathways because maybe we can turn some pathways off and basically stop some bacteria from harming us. And of course, lots of research is connected with with the microbiome which human inhabits. And that's why we can formulate that basically we can answer several questions like who is there? We can try to check based on marker genes, we can do assembly, we can do binning, and we can answer the question, what are they doing? So we can do novel gene prediction, and we can do the functional annotation. Basically, what are these genes doing in a given data set? And of course, the comparative metagenomics, it's try to find some biomarkers, maybe some correlations between cohorts of patients and so on. Before I go to the further slides, I think I would like to check the questions and drink some water.
Yes, sure. So should I read the questions or you see them?
I see from my phone.
Okay.
So does the speed of the process of discovering a new species through sequencing depend on its DNA lengths? Interesting question. I would say maybe in rare cases, but probably the factor that would influence more is how the species is widespread. Maybe it inhabits only in stream environment like volcano or Antarctic, and you need to go to a specific location to collect the species. Otherwise, you will never see it. And how easy it is to extract. This is connected to the example I gave you with this one. Because if your extraction protocol designed in a way that it will ruin part of your species just because the extraction is too harsh. So it basically ruins cell wall or membrane, and together with it ruins DNA as well, so it will be washed out. Then you will never find this species as well. So maybe another factor is cost. Also what I mentioned, like if you want to discover more new species and assemble high quality genomes, you would need to sequence deeper and spend more money. Yeah, those are the main factors, I would say, at least from what I know. Is it necessary to perform sample preparation prior to loading it onto the MinION? Yes, it's a very good question. Well, of course, you would need to extract something. Sometimes you can basically sequence cell-free DNA, which will still be in your sample. Let's give an example. We collected soil. So there will be some organisms with intact cells, which you need to destroy and get the DNA or RNA out. But there will be also cell-free genetic material, which you can sequence. But since soil is... it's not only DNA, it's lots of different molecules. You cannot simply load it into MinION because you can damage the pores, you can clog the pore and nothing will go through. And what is interesting, Oxford Nanopore Technologies, at some point they were selling a specific device for this library prep. So with this device, the whole procedure looked like you just load the sample in one device, it process everything somehow, like extract in one device, and then it automatically loads everything into MinION. But they discontinued it, I think, because it was not working as nicely as they thought. I think they still have plans to release something like this, but on the improved one. And the problem why it's so hard to do this, a very automatic pipeline, again, because it's so dependent on what kind of sample you have. Maybe this is stool sample, maybe this is soil sample, maybe this is air sample, because you can also study air microbiome, and they are so diverse, and for each of this sample you would need to do some procedures to clean it and then to extract, and for different samples you would also sometimes use different extraction kits. So yeah, the wet lab part is not so simple, I would say, but we are going there. So at least there was an attempt to make it in basically plug and play. You just plug the sample and it somehow do everything. I think maybe in a few years something like this will be more feasible. How is DNA RNA forced to go through the pore in Oxford Nanopore Technologies? I think it's because of the current difference, because we have here minus and plus, and DNA is charged in a specific way, so it will go because of that. I'm not very knowledgeable in the whole process, but I think it should be similar to how phoresis works. Sasha, maybe you're on a call here and you can comment on this, but I think it's because of the difference of like potential, I guess.
Yeah, so it initiates the potential of action, and this is what is being registered by these sensors, or these pairs.
Thank you. What is interesting, I will comment that it's cheap. The device is cheap, like a starter pack for MinION together with kits for library prep. It will cost you, I think, $1,000. But once the pores are clogged, you cannot sequence anymore. So it's not something you can reuse for many times, even though they have kits for cleaning the pores. So I think it's still a very nice option, especially for Ukrainian scientists, because you do not need to invest in super expensive equipment. You can calculate how many flow cells you need for your experiment, buy only that, spend only that amount of money, and get your sequencing done, because the protocol is still quite straightforward, I would say. Okay, I think I'm done with questions for this part. Now I will focus on a specific study we had using metagenomic methods, where we created a global metagenomic map of urban microbiomes and antimicrobial resistance, which we managed finally to publish in Cell. And it was a very long story because project started many years ago, and it's quite large scale. You can see the list of authors, and we even had a consortium as an author, which hides even more people under this name. But I think for this project, it was basically necessary. It would never have happened if not for all these people, because we were sampled mass transit system in 60 cities over 3 years, and we got almost like 5,000 metagenomic samples, which is huge. And You can try to roughly estimate the cost of the project. I mean, now sequencing is a bit cheaper, but back then maybe you can calculate $100 per sample, not including extraction cost and some shipment cost and so on. So we collected lots of samples. We targeted mass transit systems because basically public transport, because many people are there. They communicate, they touch everything, they leave their microbiome on the surface. And we provided annotated geospatial profile of these microbes we basically found across the globe. Of course, we try to do like de novo assembly and antimicrobial-resistance marker search and try to identify like novel genetic elements. But I think that strength of this lies also in the reusability. Because the data is there, you can go, you can check something specific. Because even in this paper, I mean, it's huge, it's long, but we couldn't cover everything. We had to focus on something, and it was published in 2021. And since then, we already published like several other papers targeting like a more focused area using the same data. And this is, even though the initial cost was huge, in the end, it's cost effective if you just apply new methods on the same data and don't spend on sequencing anymore. It has a very funny story how it started. So for this project, we have a huge consortium, which is called MetaSub. And the head of this consortium, Chris Mason, he is from Weill Cornell Medicine. And he has a small daughter. I mean, now she's not small anymore, but they live in New York, and compared to Kyiv subway, at least visually New York subway is a disaster. So, and he was thinking like, what if his daughter like touching everything and then just putting fingers into the mouse, like what happens? Like, is this harmful or not? And at this time, another quite famous scientists published a very interesting book, which is called Dirt is Good. And one of the comments or reviews I found was hilarious. Each answer sounds like maybe yes or maybe no. I don't know. Like the authors are afraid to take any side, which is like pure science. I think this is almost in every every good actually research, because if you claim something extremely groundbreaking without any second thought, I don't think you're being very objective. So these people are also very famous, and they work on like metagenomics. So Chris decided to check it, because as every scientist, if you have the tool, you should apply it. So, and at first the idea was quite crazy, because in many cases money goes to something more connected to human health, like cancer research or some translational studies. And back then, in pre-COVID era, if you tell someone like, we are collecting samples in the subway, it really sounded like madness. And I met Chris in 2016, if I'm not mistaken, because I was attending a summer school, and he has the very unique style of presentation. He has like 200 slides per 10 minutes. And he still makes it look flawless. And he was like, oh, join our project. We are swabbing subway. And since then I joined, and we started also to do metagenomics in the systems biology laboratory at the Institute of Molecular Biology and Genetics. So in the end, he concluded that he would advise any new parent to roll their child on the floor of the New York subway, because the idea is that you then become like more resistant to some microorganisms. I think even this phrase is now being debated, because I see many new visions in the recent article trying to rethink it. But we cannot be not exposed to everything, and I think there is some truth to this phrase. So the project actually started first on a small scale. They basically, before creating this big consortium, they did sampling only in New York, and it's very different to what we have in Ukraine, in Kiev, because we have much more homogeneous environment in terms of what kind of people live, even districts, because they found very interesting things in this study. Let's say it was, I don't know, Italian district, and people were cooking lots of pizza using specific cheese, and you would actually find some bacteria associated with like milk products and specific traditions of cuisine that was dominating this specific borough in New York, because a minority lives there. And or if it was like an Asian district, maybe they found, not maybe, they actually found some, I don't know, octopus in this samples, because people were eating more sea products. What is even more interesting, they had one of the stations flooded many years ago because there was this hurricane Sandy, and they still can find marine environment there, even though there is no water, like there is no sea inside the subway, but it still is there because we have this fingerprint. And when they discovered it, they had an idea like, what if we can actually find a fingerprint for each city in the world, and use this information to differentiate, to understand what is happening. Another striking information is that half of the samples, half of the DNA of each sample, the sequence was unknown. This is also goes back to the original question, like the first question we had during this lecture, why we had this shift in the paradigm, because previously we thought the majority of biodiversity is in the animal kingdom. We later reanalyzed this data, and this number slightly decreased because every year people add new species into the databases. So potentially you can actually uncover this, but it's still a very, very long road. And I would say it's still very expected number. So you can profile against databases only half of your sample. Sometimes it was on the extreme, like 80% of the sample is unknown, like cannot even map to anything, whether it's bacteria or something else. I mean, you can try to do them de novo assembly, and then you can try to build a phylogenetic tree and at least find some species that are somewhat similar. But for that, as I said previously, you would need like very deep sequencing and lots of money. So for this study, what is also interesting, I don't know how they got the access, or maybe it was at the time when people didn't think about it, but they were able to profile human DNA as well. That's why they could study ancestry, like what people live in these boroughs, whether they are part of some kind of minority. Because when people touch, they leave lots of skin together with their skin microbiome. It means you can actually track people. But for this large, track people, especially if they have some rare genetic disease, and it's easy to find this mutation in the DNA sequence. But for the large study, we actually, for the ethics, for the ethical reasons, we removed all human DNA bioinformatically, and we focused only on microorganisms. So we don't try to track ancestry. And this is also controversial because for every of these cities, the the rules would be different, because in some cases this is considered public space, like subway. In some cases not. I think in Ukraine it's considered like a strategical object, which is now obvious, like you can hide from Russian attacks in the subway. I think it's pretty much strategical. And sometimes it's like strictly prohibited. So we wanted to have like similar grounds between all the researchers here, and not to involve like human data in analysis. So our key aims of this consortium is basically to create this map of organism for public spaces, especially public mass transit systems, because at that point in time, let's say hospitals, they were more studied, because if in the hospital we have outbreak disease, we need to know what it is. We need to know very fast, because patients depend on that, because they are already affected in their health. That's why other places were overlooked. And since in public transport, we always have lots of people, and now after the COVID, we understand that this is quite important. We need to understand this transfer of pathogens. Of course, we are focused on antimicrobial-resistance marker detection or identification of novel biosynthetic gene cluster for drug discovery. Those are like big aims. And every year, we actually do this Global City Sampling Day, and we collect samples all over the world. Unfortunately, I don't know if we can do it this year due to obvious reasons of safety. We will see, maybe. I mean, it's safe in the subway, but still the organizations. I mean, if you do something as a public project, you still need to be responsible for people involved. And then we have a map of collected samples. It's like a global movement. We take lots of pictures. Also in Kyiv as well. Those are some old pictures I have of the team that collected samples for our city sampling day. And then basically after the samples collection, we collect them in this, not sure I have a picture. Yeah, we collect them in these small tubes with the barcode, which is unique to each tube. That's how we identify the sample. And inside this tube, we have RNA DNA shield. It's a specific liquid that preserves the genetic material, because one of the first global city sampling days, we were actually sending the samples using ice, and many of them were damaged because some shipment was delayed and it was ruined. So since then we are using the DNA RNA shield, and we can actually store and ship in a room temperature. And why we ship is because we want to sequence everything in the same facility, extract and sequence, because you create a bias. You can create a bias if you don't do everything in the right place, and it's much more difficult then to compare the results, especially even despite the fact that you're using these positive controls, negative controls, everything, then it's really hard to normalize the data in a way that it will be comparable. That's why the less factors you have, the better for this downstream analysis. Well, we found out that antimicrobial resistant genes are quite widespread in cities, because traditionally people believe they are more widespread in clinics, because people use, doctors prescribe antibiotics, people use it, more resistance appears and so on. But the city, the urban environment still can be a reservoir for these antimicrobial resistant genes. And I think it's probably quite concerning problem, because with COVID, we have one problem. It's a viral infection, and in our organism, in principle, at least, can fight it. But with bacteria, it's more complicated. And sometimes new variants emerge which are resistant to the antibiotics of the last generation. Sometimes even multi-resistance appears when the bacteria is resistant to several types of antibiotics. It's not often, but it can happen, and we don't know when it happens. So we need to be prepared, and this is another task or goal of this consortium, to understand what is happening and to understand what are the reservoirs of these AMR genes and how to monitor them to prevent something. So, of course, we found some novel bacteria and viral species, but another question we wanted to answer, what is this urban microbiome? Because maybe there is nothing novel. Maybe this is just a skin microbiome. People go touch and they basically leave the skin microbiome. Or maybe this is a soil microbiome, yeah, because people walk and they have soil on their shoes and they leave this microbiome. So we could actually compare to the public data, and we still saw that for sure, for many cases, we see lots of skin microbiome, and this will be different for different cities. So the question why it's different is also quite difficult, because I think it also depends on the planning, on the materials that were used inside the subway, because some materials are more prone to give a better environment for the microorganisms to grow, or maybe they have the higher density of population and people contact more frequently. Of course, we tested for these variables as well, and I hope I have a slide about that later. But the answer was it's actually neither soil nor skin, which was super interesting for us, because it seems that this urban microbiome is a thing itself, and it can be studied independently as in totally different community of microorganisms. And we're more dissimilar to the soil samples than to human skin microbiomes, which means that humans are a very, very major factor of what is happening. So again, it can be quite different between the continents and between different cities. So
sorry, can you go to the previous slide and the one before that? Yes, previous one.
This one?
Yes. What is here on the horizontal axis? For each city, it decreases. What is this?
No, actually, this light blue one is subway surface. It's basically a stacked bar plot. So every line shows the ratio of different sources of species, because we can have something unique to subway surface only, never found anywhere else, or we can have something that is actually from human gut, because I don't know, people lick their fingers and then they touch the surface and then they leave bacteria there. So you can try to track the origin of the bacteria, because some of the bacteria were found previously somewhere, and you can track whether it was found in skin microbiome or not.
Okay, yeah, I see. Alexander also wrote in chat that basically different samples within each city. So different coordinates where the sample was taken. I see. Just the organization confused me a bit. Thank you.
Yeah, I think I will answer these two questions we have. Are there ways to get rid of host DNA prior to sequencing to not overwhelm the reads? Excellent question. I like it. Yes, there are ways. They are not perfect. Unfortunately, you cannot remove it very nicely, and you will most probably destroy some samples. Why we didn't do it for our study is that the amount of human DNA was not extremely high. So it was not such a big deal for us. But in cases where you study human microbiome, maybe it's gut microbiome or skin microbiome, you will have extremely high percentage of human DNA, maybe 99%, and like 1% of the rest. In this case, it's sequencing everything and getting like a super small portion of reads related to bacteria is like very not cost effective. And I read some wet lab benchmarking studies trying to compare different approaches. To my knowledge, there is no like super nice method. There are some methods you can try. Wet lab, obviously, but I think there are probably some new kits from Kaigen, if I'm not mistaken, but I haven't seen the results yet. That needs to be checked in a very controlled study, probably, to see whether it removes very nicely. Maybe something like having cell culture of human cells, and you add the positive control and then you sequence, because since you know what is in positive control, you can see whether your wet lab method removes human DNA effectively and still keeps the bacteria. So this is the type of experiment I would design to check whether the kit works correctly. Is similar study possible to conduct in post-COVID era? Isn't the virus the most abundant species in all public places since its emergence in 2020? No, I wouldn't say so. I think bacteria are still more abundant, and it's very dependent on what you actually sample. Because here we were collecting samples from surfaces, and to my experience from the consortium work, if you want to capture airborne disease, you would rather need to collect air microbiome. So to collect air microbiome, you put a specific like device with lots of filters, and you basically wait for several hours until like the dust is collected on the membrane. And there you can find, I think, more viruses and more some things that are more similar to COVID. Also wastewater. I think in the wastewater you will get COVID-related things. But for this specific study, or maybe for soil microbiome, no, I don't think so. What is the traditional explanation scientists provide to explain what the unidentified DNA potentially could belong to? Those are just unknown organisms. In most cases, those are bacteria. Part of them are viruses. I don't think they necessarily belong to like higher organism. It's less rare that those are plants or something like this. And how to answer what is that? I guess the one way I suggested is that you will try to do the de novo assembly of this organism, and then you will try to do the genome annotation. You will try to understand which parts of genome belongs to genes and which type of proteins they encode. Then you can compare these proteins to what is known and maybe try to guess the function of this protein. And when you guess the function of the protein, you at least can answer like what might this organism do. Sometimes you can try to build the phylogenetic tree and try, not sometimes actually, you can try to do it every time. It just, if you're lucky, you will get a very similar organism, and maybe you can say that these new organisms belong to the same order or family of the organisms. But I think what is more interesting to actually answer the question, what kind of function this organism have and has. And for that, you would need to study it on protein level. I've heard of an oral hygiene kit that introduces a specific bacteria in your mouth for it to kill cavity-causing ones. Can something similar be done to combat AMR? Yeah, this is an interesting question. I also heard about this type of kit, and I think this is a nice application of metagenomics in dental practice, and I hear people try to do it more and more. I think actually in our institute there is a study actually like focusing on the dental procedures and the oral microbiome. Can something similar to be done to combat AMR? People try to do it with phages because this is a virus that infects bacteria and kills it. This is like an alternative way to try to kill bacteria instead of using like typical antibiotics. I'm not sure how successful is that. I don't think so. Maybe this will be possible later, like to have a mix of phages which you can, I don't know, eat or drink, and then they start killing bacteria. Maybe something like this. Okay. Let's go further. So this one we saw. Yeah, this is actually a profile of AMR genes with resistance to particular drug classes, because as I mentioned, first of all, we have drugs of different generations, and you know, maybe even on your personal with your personal experience that like a few years ago you were prescribed these antibiotics, and maybe nowadays people don't use it anymore, and you are prescribed for the same thing with different drug. So that's why we have different drug classes to basically also target different types of bacteria. And we checked whether like we have a specific concentration of bacteria resistant or to a specific drug. I mean, here we actually check against databases, against specific genes that we know that they are resistant. To be honest, this is based on a dry lab analysis only. And to prove that something is really pathogenic and is really dangerous, you would actually need to test it additionally with the drug. For that, you need to have the living bacteria, which is sometimes hard to get because of the problems I mentioned before, because you cannot always culture everything. But even this dry lab analysis, this pure bioinformatics, is quite insightful, because we need to understand whether this changes through the time, whether there is some kind of dynamics, whether it's consistent. Maybe we will always have the same profile, or maybe we, I mean, we did not include it in the final manuscript, but we had some data on trying to correlate the proximity of collected materials to the hospitals, because we know that hospital can be a reservoir for the AMR genes, and it worked for some cases for some Asian cities, but it didn't work for some US cities. That's why we could not find out the exact reason why, so we did not include it in the manuscript, but with better resolution of data, maybe we can understand. It's definitely depends on the regulations of antibiotics usage in a specific country, because if it's not very controlled, then probably you can see the correlation. And maybe still wastewater is better type of sample for that, but even with the surface sample we could identify in some cases some correlations, because people go out of the hospital, maybe they touch everything, maybe they are like still sick. Then
sorry,
yeah.
short question, does the color on the right graph indicate anything, or is just
actually I don't remember. I think no. I think it's just to differentiate between, or maybe this is actually, maybe it could be a continent, actually. Like the green, it looks like
yeah.
Europe.
Looks like that. The red is the North America. Okay, thank you.
Yeah, green at least looks like Europe. I think it's a continent. But I was talking mainly about applications, but you need actually quite a complicated machinery to be able to analyze this amount of data, because almost 5,000 samples, I guess at least 2 million reads each, sometimes more, depends on the sample. So you have huge data set and you need to do something with that. And you need to perform such operations as like search or maybe comparison. And for that, one consortium member from Zurich, they developed a tool for fast DNA search alignment and assembly. It's actually, my student knows more about it because he took part in developing some specific functions for this software. But the idea is to build a huge index of all the sequences in the database using the so-called k-mer concept. K-mer is just a string of the length of k. And you can build a graph of these k-mers, and the nodes have edge if the k-mers have like overlap between each other. The operation, like the cost of building this index is quite high. You will spend lots of computational resources to build that, but once it's done, the search is quite fast. So you can actually try to go to this link. Maybe I can copy paste. Let me try. Yeah. So with this tool, you can insert in the search field a DNA sequence, and then it will search again the whole metasub database and tell you if there is anything in this world we found in our data set that is similar to your sequence you inserted, which is nice. Maybe it partially answers the question we had in the chat, not in the chat, but in the Slido, how to answer like what is in the sample, what is this unknown DNA. Let's say you're like independent researcher, not connected to Metasub in any way. You found something which is unknown, you can insert in our database search, and you can at least see whether someone else found something similar in which environment and what kind of country, what kind of city, what kind of, I don't know, temperature conditions and so on. It might give you some idea how to understand what is in your sample. We also tried to actually infer microbial signatures for each city and understand whether we can actually identify and compare cities with each other, or blindly say that, oh, it was, I don't know, Paris. And it actually worked quite well. I think this was a simple random forest classifier here. So once we increase the training set size, and you remember we have almost 5,000 samples, the accuracy is almost like 90%. And then we also try to estimate whether accuracy is dependent on a specific metadata variable, because we have population, surface, elevation, whether the city coastal or not, population density region, this is surface score, surface fine, and then average like temperature in June and what kind of climate. And interestingly, it's not uniform across the city, because for some cities, like everything works. For some cities, only some variables are more like influential. For example, this is second is Zurich, and we see that population is really a factor that correlates with the microbial signature we found for this city. There is a slight problem maybe with this plot, is that we didn't have actually uniform amount of samples per each city, and I think it in the end might have influenced some results, but I guess it's still the first study like on this scale where we tried to do it. Actually, I later found like recently papers studying this in more details using our data, and I'm very happy about that. It means our data was not stored somewhere and forgotten. People actually reuse it, so it was quite interesting. We also work on some optimization and automatization, because to publish this paper we actually had to develop a pipeline to analyze data, and we had several versions of this pipeline, and this one I'm showing now, it's actually was developed after we finished the paper, because while we were doing it, we understood that it's not flexible, it's hard to add new modules. It was based on Luigi. I don't know if you heard it. It's also workflow management system. You heard yesterday, I guess it was very nice talk from Ton from Wellcome Sanger Institute, with whom we actually collaborate on imaging, but it's totally different story. But he like highlighted this importance of using a specific environment where you do computations that is reproducible and fault tolerant. And he mentioned that there are many pipeline management systems like CWL, Common Workflow Language, SnakeMake, Nextflow. So for the first version of the pipeline, we use Luigi, which is pure Python based, and at the moment we use SnakeMake, which is also Python based. And I mean, it looks cool because the idea is to make a very predefined module structure. So we have a template repository on GitHub. You just clone this repository, which gives you already structure of your model, and then you just basically change it for your specific model, which can maybe do the phage hunting. So, which means then that people can use your model independently and use the output of assembly step as an input for your phage hunting. So the system is modular, and then you can connect these different things. And it's supposed to work super easy, because you basically clone GitHub repository, then you create Conda environment, and then you activate, and then you run some tests. Unfortunately, it's not always so easy on practice, and some of my students were recently developing additional module for camp, and we encountered some Conda-related problems, which is almost always with the software versioning. We managed to overcome that, and I still think it's much better than what we had previously, and I'm sure we will try to make it even better. I mean, when we have lots of people and people can contribute to open source software, you can actually create something quite nice. Then, yeah, we also have MetaSub Europe Society. We basically established recently, because for some operational things, it's nice to have a legal body in Europe, because the current head of MetaSub is in US, and we try to even more expand our activities. And I would like to highlight another study, actually, which was actually a continuation of MetaSub ideas, and it's a little bit different approach. It's metagenomics in forensics. I was donated slides from my colleague who is actually here ahead of our no, I think he is vice director of our MetaSub Europe society. And this is a project we did together. So as I mentioned, first of all, you can track people using metagenomics, but this is not what the project about. We actually not using any human DNA. Again, not exactly for ethical reasons, because forensics operates on the laws of a specific country. If we try to use the results of the research in investigation, and the idea was different. What we found out in MetaSub, if we can go maybe for this picture. So we collected samples in many different cities, and when we tried to infer these microbial signatures for each city, it worked nice, as I showed you. But if you add something new to the data set, which was not profiled before and which is not similar, it fails to, it's much harder to identify. Let's say maybe we collected something here in Africa. It will be easier to answer the question whether this sample is different. Let's say it's different. But what we want to answer, we want to get the actual location, that it's somewhere in Africa, because maybe it's more similar to the Africa. And when we opened this data, we used it in the open challenge. We hid information about some metadata for some samples, and we asked people who took part in this challenge to guess the mystery sample. And it was extremely hard. And we then understood that probably the resolution of this collection is not high enough to be able to approximate then a location of the new mystery sample. So the idea was to focus more on a single country, and in this case it was done in Poland, but we continued the ideas we had in MetaSub, that we can actually infer a specific unique signatures of different places of collection. We already know, and it's quite obvious, that the climate is very influential on what you will find in your sample. That's why the country was divided in like several climatic zones, which are depicted on this map. And we tried to collect samples in all these climatic zones to see whether we have some kind of pattern. And the idea is that people leave fingerprints, and with time they fade, which means if you touch something and we can profile it quite soon, we can kind of answer the question whether you touched it. But this is connected to skin microbiome. The same can be applicable to soil, because this project was more focused on soil microbiome. If you have a soil on your shoes and you came from a specific place and you left this soil, someone can then collect this, do metagenomics, and basically identify the profile of the soil which was on your shoes. It sounds futuristic, but it's I think it's already more or less possible, I would say. And actually it worked even better than we expected. It was a huge also study, and it's a consortium of many different bodies. I mean, the universities from Poland, also actually software company, and this is, I think, quite nice that industry can also join such projects sometimes, because industry operates on different standards and they have different workflow development workflow adopted, which we lack sometimes in research. So I think it's quite cool when you can basically combine expertise from academia and the industry. So it's quite a crazy study when it comes to the data generation, because it was actually deeply sequenced, because we wanted to create the panel later on of a specific sequences that would significantly reduce the cost of the forensics tools we wanted to create. And later on allow us not to sequence every time, but just to use a very dedicated panel and sequence what we think will be able to differentiate between different places. Of course, we collected tons of metadata and weather data and some physical-chemical analysis of soil samples, and we did it over 4 seasons with 80 locations and 3 samples per location. So you can imagine how much data we have, and this is on the raw data, and on each step of analysis it doubles, because you want to preserve the intermediate results, so it's quite challenging later on for the computational analysis. And what I don't think I can tell you much yet about this because it's yet to be published, but what is interesting here is in the final panel we had lots of unclassified sequences, which means for the purpose of identifying whether some object came from a specific place, we actually don't need to answer the question what kind of bacteria were there. It's enough for us to know that this specific sequence was there. And this is why we were able to utilize even the unknown information. It is a very specific case for human health application. Usually you want to know what is in the sample. But in this case, if you want to basically track if this piece of soil came from the mountain region, you don't need that. And this is actually possible with the tool I showed you previously, this K-mer-based tool that tried to assemble everything into the K-mer graph and then do some operations over this graph, because it doesn't care about annotation of the species. It just cares about the sequence and about our genetic code alphabet. And that's why I think usually for metagenomics, you would apply a combination of tools, annotation-based, when you take the database and try to check against reference organisms, and then annotation-free tools, which just operates with the raw sequences, basically, and try to maybe find some frequencies or peculiarities in the sequences itself. I have a few slides, just general slides about my antibiotic resistance. I just want to thank again my colleague who provided me with the slides to tell you about this project. And let me check maybe questions.
Yes, there is one new question.
Yes? Yeah, I see one. What are the ways to address imbalanced strain datasets when it consists of manually collected samples, apart from collecting more? Bootstrapping. Yeah, I think we did some bootstrapping. It's not perfect, I would say, because imbalance sometimes was quite huge. For the study, we had certain budgets, per city, but in some cases, some cities were able to acquire additional funding and then sequenced more or much more. And that's why the discrepancies sometimes were high. Yeah, I think I cannot come up with something else than bootstrapping now. Another thing actually we did in the paper, it was not a bootstrapping on the level of samples, but we were subtracting part of reads per each sample and testing how methods behave if we slowly increase the number of reads we add. And so you can try to balance it on the level of number of reads per sample, actually. So you can, in some cases, just decrease it for other samples. And basically you randomly select, and then maybe you can increase and see the trend if something happening. This, yeah, I hope it somehow answers the question. I think we still have lots of time. These slides are rather general ones, again, to explain how this antimicrobial resistance behaves and how it emerges. And I think this is rather a common knowledge, and you have, you know, many of you know this, that if we try to kill bacteria with antibiotics, we might encounter a situation when some bacteria basically survives this. That's why if you are prescribed to get drugs for 5 days, even if on the fourth day you feel much better, you still need to drink it for all the prescription period, because you never know if you're not killing everything till the end, and maybe something survives and goes further, and then you basically acquire the resistance. Some of the colleagues from the consortium, they were working with patients in the clinics for cases when they acquire the resistance. And sometimes they sample patients before, just to test prematurely, like in advance, if something bad might happen. And they saw cases when they basically sometimes get this resistance in a few days, like they sampled this patient today, and in 2 days he or she already have this resistance. So it can be quite the rapid event. But as I mentioned, we have plasmids, the circular DNA that can be transferred basically between bacteria, and it's so-called horizontal gene transfer. And if the plasmid contains this resistant gene, it means that this bacteria also will have resistant genes. That's why in the community of bacteria, even though they are not necessarily from the same species, you would still find increased incidence of the same resistant genes because they can basically transfer between each other. That's why it's so dangerous. Yeah, as I said, usually you would actually check against databases. And the problem is that the information in databases is extremely limited. So we need to find a way how to find this resistance de novo. And I think this information is extremely crucial, but it requires very controlled, sometimes simulated environments, sometimes dangerous because of that. Let's imagine like you have different bacteria and you want to test how fast they acquire the resistance. You would actually need to create this resistance bacteria. And if your safety level is not good enough, it might result in some unfortunate events, I would say. And I will advise you to actually go to YouTube and search for this video, which illustrates how resistance work very nicely. So they had a huge table separated in these lines, and in each strip, they added a specific amount of the drug, of the antibiotic. And toward the center, they added more. So here in the center, they added like a huge amount. And it didn't matter how much they added, because in the end, it's only the process of the question of time. The new populations were emerging every time that developed this resistance, until they started to grow even here in the center. But this is a very extreme case, because the truth is the cellular machinery inside the bacterial cell that tries to resist the antibiotics, it's very energy consuming. And if you stop administering the antibiotic to a specific place, then usually bacteria can lose this resistance. So it's not only a one-way process, and it's sometimes the opposite sometimes can happen. That's why you can even say that sometimes overclean is bad because if you try to apply the same chemical or drug to the same thing, it can eventually develop the bacteria. There was a study where they actually do the metagenomics of a sponge you use to clean dishes in the kitchen, and it was extremely dirty in terms of like bacteria they found there, like worse than the toilet seat or worse than anything else. So they are basically, their conclusion was to change it as frequently as possible. So do not use it for quite a long time. And that's why, yeah, it's sometimes interesting to understand that our knowledge was totally flawed because before all these methods were developed. I think this is all from me. Maybe I'm faster than I was intending. Do we have questions? I don't see in the chat.
Thank you, Alina. Very detailed talk with many aspects, actually. But yeah, at the moment I don't see questions, but a lot of people.
Yeah, but we answered
me including,
questions.
yes, we were asking them during the talk. So maybe someone will ask extra questions now.
I see some questions in the chat here, actually.
No, those were just comments.
Okay. Yeah.
I actually, I didn't want to distract you too much from the talk, but since while we are waiting, can you share the screen again? That graph that I was asking, the question about distribution and the way of sampling the cities.
Was it here or here?
No, actually, I had several. I have a bunch of questions. Let's start from here. For example, these are different points in the city. But again, the question of same representation. Like I can see that in Hong Kong, you have lots of data points. But for example, in Denver or in New York even, it's less. So the question is how do you average them? How are they distributed evenly? Because from these graphs, yes, Hong Kong looks rather different from London. But then again, maybe London was all taken from one small part of it, or mostly from one part of it, and then Hong Kong is more evenly distributed and there are more points. So that question, how to compare them.
I don't remember very specifically for this paper because this is a follow-up of the main paper, but in general, we tried to do a fixed study design, so we will repeat the same sampling places over the years. It's hard to maintain, to be honest, because in some cases the team changes for the city that is sampling, and you need to basically negotiate with people again and make sure they collect from the same places. And sometimes, as I said, people can add their own budget for sequencing and they can afford to sequence more. In this case, we see that clearly in Hong Kong, we have more. So I would rather either subsample here or in this case, we are trying to see the distribution of microbiota sources per each city. So I would go for average per source. Let's say we can calculate for Hong Kong that, I don't know, 30% of microbiota found in for this air microbiome in the public transport system comes from human skin. So you would average in this case per type of the source, which is, I think, okay, as long as we don't have the situation when here is 3 samples and here is 100 samples. So as long as the difference is not extra huge. And sometimes you don't necessarily want to compare, but rather to study the trend per each city. Because it seems that for some locations that the source, like the human skin, was more than the rest. Then you would go further and study the metadata associated with each sample and check whether there is any correlation with type of surface. Maybe something is made from wood and something is made from metal, and wood is better for, I mean, here's air microbiome, but I'm talking in general. So this is a trade-off between diversity and reproducibility, because let's say you collect your sample always on the, if it's a subway, you collect on the platform. What would you like to do? You want to collect more stations, but always collect on the platform, or you want to cover more places and you collect on less stations, but you collect on the platform, on the stairway, on maybe on where people buy the tickets. And you need to, again, calculate how many samples you need per group. I would say at least 5. And then how many groups you would have and how much will it cost per sequencing. So it's hard to design. And I think we were not always optimal in this, especially in the first years where the collection was a bit more chaotic, because we were like, oh, let's collect everywhere and everything. And then we realized we need more controlled study design.
Yeah, it just looks for me that it's a different, difficult task by itself, the data acquisition and data collection.
Yeah.
Because even for New York, like the examples you are showing, you distinguish different parts of the city which have their own characteristics, and then like how to like allocate the weights properly, like to distinguish like different parts of cities, or if you average them, like, and there is like some city region where more of those marine organisms are present, for example, and like how to weight it properly, basically. I see there is one more question from the audience. What were the results of the study where patients' microbiome was sequenced and some of them later developed antimicrobial resistance?
I don't know. I hope they survived. I don't know, honestly. It was the study of a colleague. I know some stories from my other colleague. He's also part of the consortium. He's from Tübingen. He's rather from the medical field, or like a very applied research, because they have huge hospital in Tübingen, and he's more into cancer research because they use nanopore sequencing to study like the structural variations. But sometimes in the hospital they have infection spreading, and they need to do super rapid sequencing to identify what exactly is the source of the resistance. And if you don't do it quick enough, people really might die, because it might not be harmful for a regular healthy human. Just give antibiotics and you will be okay in 3 days. But imagine a whole ward of patients with cancer who are restricted in what they can take, especially if they are on chemotherapy. So it can be really dangerous. That's why this culturing of bacteria is not an option for such cases. You need to swap different surfaces and eliminate the source of infection. I don't remember the exact case, but they really struggled at some point to find exactly what it was, whether it's maybe spread from the kitchen, or maybe the source was something else. So that's why the response should be quite fast, and for that we need to develop fast sequencing protocols and fast analysis tools. Yeah.
Maybe one more question, rather short one, from the Polish project. We were showing the map, and
this one.
Yes, this one. You said that these different clusters, that they correspond to different climatic groups, right?
Yeah, somehow,
but
I guess.
Are they, if you know the details, was it like some sort of clustering algorithm where you, based on what is observed, it was just corresponding to that, to this or that climatic zone? Or what was like the process of assigning these climatic zones,
yeah, actually,
or clusters?
Quite,
because it looks very strange that you have a lot of like yellow, and then suddenly there is like random blue dot close to the Ukrainian border, or for example pink dot closer to the Czech border. So this heterogeneous spots, or like even anomalies, like why is that?
Some things are easy to explain without any difficult analysis, because here they actually have mountains. And here it's like more a seashore, but we still have some things that are a bit more random.
Yes.
And it was based, it was not me who did it, but it was based on open weather map. And we took into account many different parameters, like average temperature in winter, in summer, like precipitation, I think some trends.
Okay, so it's purely independent data from the data that you were like obtaining. It's just
yes, actually,
geographical.
Yes, it is. It is independent, and we did some kind of clustering when we collected information from here. So it's not that we visually selected the clusters because we felt like it. No, we had to do a decision which would be based on some kind of data, and you can then later do other way around. Once you get the metagenomic samples, you can try to check whether they indeed correlate with.
Yes, that was my next question.
Yes, you can do it. Yeah, and yes, it is.
Okay.
And actually, there are many different types of correlation. What I saw when I was checking data also, that obviously it correlated whether it was collected in an area that was somehow processed by humans or not. Like whether it's wild nature or not, it's like extremely different. Also, in some cases, these dots are more closer to some reservoirs with water, which affects the microbiome tremendously also. And in one case, we had constant misclassification of a few samples, which we could not explain first, but then later we realized that it was collected from the polluted soil, polluted with some heavy metals, because it was not far from some huge factories.
Okay, thank you once again. I think we can stop here.